Hi, today on Viva Bites we are talking about the RNA interference pathway. So my main sources for this are He and Hannon 2004, microRNAs, small RNAs with a big role in gene regulation, and also Wikipedia. Firstly, I'll talk briefly about what it is, why it's evolved, give you a very short overview, look at the differences between miRNA and siRNA, and then go through a little bit more detail for the various steps involved in RNA interference. So, what is RNA interference? Well, it's an evolutionary conserved sequence-specific gene silencing mechanism that is induced by exposure to double-stranded RNA. It's mediated by two types of small non-coding RNAs, microRNA or miRNA and small interfering RNA or siRNA. Specifically the second part, but they're both so close that they can both trigger the pathway. Why did it evolve in the first place? Well, it's thought that originally it was a defense, still is actually, a defense mechanism against viral infection or parasitic nucleic acids. But it's also got other roles, so it's also involved in regulating protein levels at the translational level via mRNA destruction or translation inhibition. You also see that it has a role in epigenetics, so in particular in plants it's important in sequence-specific promoter methylation. In fish and yeast it's crucial to heterochromatin formation, and in worms it's been shown to be important in transposon silencing. So how does it do all this? Very short overview. To initiate the RNAi system, you need a double-stranded RNA, which is called the trigger, and it's about 500 base pairs long. This trigger is ultimately processed in vivo into small double-stranded RNAs of between 21 and 25 base pairs in length, and these are designated as small interfering RNAs or siRNAs. One strand of this siRNA duplex is then selectively incorporated into an effector complex called the RNA-induced silencing complex, or RISC. And finally, RISC then directs the cleavage of the complementary mRNA targets, a process that has also been called post-transcriptional gene silencing, or PTGS. So I've been talking interchangeably about miRNA and siRNA, but what's the difference between them? Well, let's start with miRNA. They are generally between 21 and 25 nucleotide long, non-coding, and they are derived from larger precursors that form a very imperfect stem loop structure. So you've got a sort of hairpin, imagine a hairpin, where you've got the nucleotides on either side that join together, and then the loop at the top. But it's imperfect in that you don't have necessarily a one-to-one correspondence exactly. So there'll be a couple that form a little tiny loops here and there where there is no corresponding nucleotide. So the mature miRNA is often derived from only one of the arms of this precursor hairpin, and it's released from the primary transcript through processing via two ribonuclease 3 enzymes, or RNase 3. The first one's called DROSHA, the second's called DICER, and at least in animals, plants is a different matter, but in animals, most miRNAs bind to the target 3' UTR, untranslated region, with very imperfect complementarity, and they function as a translation repressor. siRNAs are usually between 21 and 22 nucleotide long, also non-coding. It's usually a perfect stem loop structure, but and has a very slightly different mechanism. So the similarities. They both have very similar molecular characteristics, a very similar origin and ways of acting. So both have to be double-stranded and are between 20 and 25 base pairs long. They both share the same second processing enzyme, the second RNase 3 called dicer, and are closely related, have very closely related effector complexes, so the risk is very similar. And both act to affect post-transcriptional repression, either by triggering the RNA interference pathway. Now, differences. One of the main crucial differences is the molecular origin. So miRNAs are produced as a distinct species from a specific precursor that is encoded in the genome. The structure of the primary miRNA transcript and the recognition of this processor by a nuclear processing machinery probably determines the sequence and structure of mature miRNAs, i.e. how thermodynamically stable each strand is relative to the other. That'll determine which one's loaded onto the RISC. In contrast, small interfering RNAs are sampled a bit more randomly from long double-stranded RNAs that can be introduced exogenously or produced from bidirectionally transcribed endogenous RNAs that then anneal to form the trigger, the double-stranded RNA. The second main difference is the mode of target recognition. So miRNAs usually bind to the target's 3' UTRs with imperfect complementarity at multiple sites and negatively regulate target expression at the translational level. In contrast, siRNAs usually form a perfect duplex with their target, so exact matching at a single site, and therefore are able to directly cleave the target mRNA at the site of complementarity. Now, that's all well and good, but actually recent findings suggest that these differences may not entirely be universal, and the distinction that we make between miRNA and siRNA might be more based on the order in which we discovered them and sort of how we discovered them. And in fact, there's much more overlap than was initially envisaged. So, for instance, the original distinction that you see between siRNA and miRNA is that siRNA has perfect complementarity and cleaves the mRNA, whereas the miRNA has an imperfect complementarity and usually prevents translation rather than destroys the mRNA. However, the extent of complementarity might be what determines the mechanism of silencing. For instance, all characterized plant miRNAs have a near complete complementarity at a single site, either in the coding region or in the UTRs, and the result is that their target mRNAs are destroyed by cleavage. So that's the plant. So in essence, the plant miRNAs act like animal siRNAs, right? There is one exception. MIR172 acts like a translational repressor, so the traditional animal miRNA. And there is one mammal miRNA that acts with perfect complementarity. So that's the MIR196, and it cleaves HOXB8, so it also acts like siRNA. And then conversely, when siRNAs have imperfect complementarity, then you get translational repression rather than cleavage. So there's less of a difference between the two than was initially thought. So I'm going to go and talk about both because once they've been past the generation, once they've been generated, they've triggered the RNA interference. It's very similar methods, so I'll cover both together. So let's go over a bit more in depth the various steps that are involved in the pathway.